introduce our last speaker. So Jennifer Kuzma is coming to us as the Goodnight and NCGSK Foundation Distinguished Professor in School of Public and International Affairs at NC State University. And she's also the co-founder of the Genetic Engineering Society Center. So thank you.
Well, I want to thank the organizers for inviting me. I've really learned a lot this afternoon. And I'm glad somebody asked a question about genetic modification because that's my specialty. And just starting to learn about the microbiome and what might be done with it. So I'm going to take you on a late afternoon journey, thank you for sticking around, on the societal implications of microbiome engineering. And the way I'm going to start is first looking at the challenge of the diversity of the ways you can engineer the microbiome, and then moving to the first generation of genetically engineered products, which we have a history of, and we can learn from that history in order to address the future of microbiome engineering. And then we'll move on to microbiome engineering and uncertainties and some societal issues. But again, I'm just learning about this area. I look forward to learning from all of you. And then some ideas for moving forward in a better direction than we did with the first generation of genetic engineering. So the engineering microbiomes is obviously a complicated subject, and it can be done chemically, as you all understand better than I do, with metabolites and activators and suppressors. But it can also be done by changing the cellular composition, which is in blue here. The green is more of the chemical methods. What I'm going to focus on mainly today is the engineering of DNA of the microbiome, either intracellular or externally adding phage methods of engineering cells or CRISPR-Cas9 systems, for example. So you have not only a diversity of ways that you can engineer the microbiome at a macro level by strategy, chemical, cellular, or genetic, or all of the above, but you also have a diversity of ways if you're going the genetic route of engineering the microbiome. And synthetic biology, where we have different transgenic varieties, transgenic bacteria, or CRISPR-Cas9 systems that are used to engineer the microbiome in situ, or even new and novel DNA-based systems from synthetic biology, maybe even artificial cells and artificial cellular systems, or alternative codon systems. And that's the way synthetic biology is moving, and it might be a little bit ahead of the microbiome engineering field right now, but it's coming, and it will be possible. So you have this really huge challenge of the diversity of different ways and the diversity of different DNA modification methods that you can use, which really presents a really big challenge for regulation and governance. In fact, it's very difficult to talk about the societal implications of all of microbiome engineering. But I'm going to try by first giving you my definitions of regulation and governance. When I'm talking about regulation, I'm thinking more of the mandatory kind of hierarchical top-down processes that have legal formality to them. So things like laws and rules and the way that the federal agencies actually gather data from people who want to put a product on the market and then review that data and make a decision. It's a really interesting space in science and technology policy because it's where data or the rubber really hits the road of decision making. And that's been my expertise for many decades now, is how do we take complicated scientific information with a lot of uncertainty and make a decision with it. However, when you talk about oversight, it may be more than just the top-down formal legal ways that you govern or regulate a product. And that might include things like voluntary standards, data sharing, public engagement, corporate social responsibility or responsible innovation, which we're calling it more and more these days. And then you have something that's even broader, which is governance, which is essentially how we care for these technologies as a society, all the way from the birth and the research stage to the consumer use and the marketing of the products. So we all have a responsibility for governance, although oversight and regulation might be construed more narrowly. So how have we been regulating the products of biotechnology? There's something called the Coordinated Framework for the Regulation of Biotechnology. And it's been in existence since 1986, and it largely still operates as it was envisioned in 1986. This was a policy document that came out of the White House, the Office of Science and Technology Policy. And the principles were that there are no new categories of risk from biotech products, and therefore we don't need any new laws. And it's the product that should be the focus of regulation, not the process by which that product is made. So in other words, conventional products like microbiomes that are not engineered, but you might be introducing through a probiotic, are not any more or less risky than genetically engineered microbiomes. But the risks may be greater or lower for either one, conventionally or genetically engineered. And so therefore, we don't really need to devise a new law. And that we should focus on the characteristics of the product, not the process by which it's made. For example, through conventional breeding or compilation of existing microbes versus recombinant DNA or molecular engineering. And what this policy document did is it took existing laws that in some cases had been around for decades, and in some cases almost a century, like the Federal Plant Pest Act, the Food, Drug, and Cosmetic Act, the Pesticide Act, Federal Fungicide, Rodenticide, and Insecticide Act, and the Toxic Substances Control Act, and told the federal agencies to, okay, now use these laws in order to regulate the products of biotechnology and promulgate rules under these laws. So you got some really kind of strange interpretations of these old laws in order to fit the new products, the new technology products. For example, we've mainly governed plant biotechnology or genetically engineered plants through the Plant Pest Act. Because molecular biologists, when they were first making genetically engineered plants, used to use plant pest sequences from Agrobacterium in order to get the DNA in. Now that they're no longer having to use those sequences with CRISPR-Cas9 systems or other molecular engineering methods, those gene-edited plants are not coming under those laws or those regulations, and most of our gene-edited plants will not be overseen and on a pre-market basis by the Department of Agriculture. Another strange kind of interpretation of law to these new products was the Food and Drug Administration exerted authority for genetically engineered animals under the Food, Drug, and Cosmetic Act. So they were to regulate genetically engineered cows or salmon, for example, which is coming onto the market very soon, as new animal drugs, with the idea that the gene that you're putting into the animal changes the physiology of the animal and therefore acts like a drug. So we can capture that product under that existing law. Most of the microbial products will come under the Toxic Substances Control Act with EPA, the Environmental Protection Agency. And that is interpreting genetically engineered microbes as toxic chemicals. So you see these kind of strange interpretations of legal authorities to pace with the science as it evolves. But again, no new categories of risk was the principle, therefore no new laws, and it's the product, not the process. So with EPA and genetically engineered microbes under TSCA, it's even more narrow than that, in the sense that it has to be coming from the gene has to come from a microbe or an organism that's of a different genus than the organism that you're putting it into. We call that intrageneric. It only applies to new commercial microbes or commercial activity. So most of our regulation in the United States is based on interstate commerce. And so therefore, if you're not using genetically engineered microbes commercially, like the DIY or do-it-yourself biologists, it may not need to be regulated. If you're not selling it to your neighbor, but you're giving it to your neighbor. And so as biotechnology is becoming easier and easier to do in makeshift labs and garages and communities, the meaning of commercial becomes more and more important for regulation. And furthermore, a genetically engineered microbe doesn't come under TSCA if it falls into another category, like plant pests, drugs, dietary supplements, foods, etc. And there's very little post-market monitoring required. And furthermore, the burden of proof has generally been on the regulatory agencies to show unreasonable risk, as opposed to showing a good indication of safety. Now, the National Academy of Sciences came out with a study in 2017, and in full disclosure, I was a part of the committee where they indicated that with increasing product complexity and novelty that microbiome engineering presents, we're going to really be at a loss with regard to this existing coordinated framework system. And even though the risk assessment endpoints won't be new, in other words, we'll still be looking at human health outcomes and ecosystem outcomes, the pathways to those endpoints have the potential to be very different given the complexity and the diversity of microbiome engineering. Furthermore, the just sheer volume of the number of biotech products that we're going to be coming down the pipeline are going to challenge these regulatory systems that are often underfunded, understaffed, and under attack by not only Congress, but NGOs and the public. And so to enable effective regulation, it would be beneficial to have a single point of entry for the coordinated framework, which we have not had, and we still don't have. So this is the model that we proposed, is that the entry point for this coordinated framework, because it is so complex, because we're still trying to figure out how these existing laws are going to cover some of the new and highly convergent products, you'd have an entry point that then, depending on the novelty and complexity of the product, you'd have a more or less traditional regulatory system. So the more traditional way that we operate in the United States is really the decisions are made by the federal agencies in partnership with the industries or the companies that are submitting the product. However, as you get more and more unfamiliar and complicated, I don't think you can see this, but the right side of the slide here, there's more of a rationale and more of really a duty to engage external and affected parties as well as citizens and expert advisory panels. Because we haven't been faced with these products before, we need a more rigorous, socially robust process. So that was the recommendation. And that's where we move more from kind of traditional regulation on the left-hand side to governance and societal governance, where we engage the publics, we engage interested and affected parties, and outside experts to make these decisions, not only the technological elites, which I guess we are all a part of in this room, to make these decisions. So that's one important point, I think, with microbiome engineering, is that we could begin to think of systems which we would devise in order to do a more rigorous job and prevent the public failures of the first generation of genetic engineering. So what are some of the public failures of the first generation of genetic engineering? Mainly done in plants and mainly with just a handful of traits like Bt and herbicide tolerance. We did a multi-criteria evaluation of the oversight system for GMOs several years ago, but I think this still really holds up today. In that the good part is that it's highly flexible, so it's allowed it to change with technologies over time, for the most part. However, it has a weak legal grounding. So as political pressures kind of oscillate over time, the system has also come under those political pressures to become either more or less rigorous over time. It's extremely complex in the institutional structure, and it's very hard for small product developers to navigate the system and can be very costly at times. But more from a public policy standpoint, in a public rigor standpoint, there's very little transparency on the part of to the public, a very low level of informed consent. In medicine, you have maybe a more direct informed consent process, but in food and agriculture and environmental biotechnology, generally these products are put on the market without most people knowing or understanding, and in some cases consuming them without knowledge or consent. Few opportunities for public input, and again, low capacity of the regulatory agencies that are very often underfunded. So in summary, the first generation, a highly contested political environment, a lot of controversy, delay, rejection. We're seeing more and more people avoiding GM foods, seeking out organic or non-GM labels. Governance decisions were open to really only a small set of technical experts. But we also had this mantra of sticking to the sound science, which is in regulatory terms has been interpreted as code for direct human health and ecological risks. So it marginalizes a lot of people's values and worldviews, and so whose values count? And so in summary, I think that they've been a market success, but in many ways a public failure. So some of the questions and some of the reason why this public component of governance is so important is that we all have different ideas about what a harm is to us. And so who gets to decide what a risk is or what they care about? Our values are embedded in what we care about in a regulatory or oversight system. Who gets to decide when there's enough information about risk? There's almost always uncertainty, especially with regulatory safety studies and risk science. And who gets to define what level is safe? So for example, here's a dose response curve. Usually our data is at high concentrations where we're feeding a lot of the product to rats or other laboratory animals and looking for a big response. But what we really are concerned about with food risks are more low-level risks, chronic over a lifetime of consumption. And so if you don't have good data, drawing the line poses a lot of uncertainty as to a dose response curve. But even if you have perfect data, what's safe to me may not be considered safe to someone else. So in other words, where do I set my regulatory standard? Is it, right here, or right here, or right here? Safety is a value judgment. So values come into play with safety. It's not a purely sound science concept. Furthermore, scientific uncertainty is interpreted according to worldviews. Industry studies are not trusted, but that's the basis of regulation. And studies showing any risk in the literature have historically been discredited by biotechnology developers in particular, in the agricultural space, not so much medicine, but. And then also biotechnology developments have made the argument that we shouldn't be regulating biotechnology at all. So if the risks are the same in kind, and it's the product, not the process, and if it's strictly based on sound science, the two logical conclusions are to regulate every product, whether it's genetically engineered or conventional, or regulate no products, because conventional breeding and conventionally derived products are not often regulated in the agricultural and environmental space. But which one you choose is a matter of worldviews. So I've always made the argument that you cannot design a regulatory system strictly based on sound science or science. It's a matter of values and worldview. So now a little bit on the risk perception and what does influence our conceptions of safety. One of the models out there in the social science literature is called cultural cognition. And so it really depends on our position and our worldview from a scale of hierarchical to egalitarian or individualistic to communitarian as to how we perceive risk. And this is Dan Kahan from Yale, and many others have studied this kind of model. And we find that people in this quadrant often perceive the risks of technology as higher across a lot of different technologies: nuclear engineering, pesticides, genetic modification, and so forth. So that's one thing that influences people's worldviews. The other is trust. The other big one is trust. So we've done some studies on public perception of GM foods, and many others have as well. And we find that one of the biggest correlations, significant correlations, is trust in government to manage the technology. Other factors are features of the technology itself, which I don't have a slide for, such as whether you can control it and whether it's familiar to you. So here's the divide in summary from history. The divide is what the public cares about, which is transparency, mandatory systems, opportunities for input, information, and choice, and how we've generally operated in the agricultural biotechnology and environmental biotechnology space. Little transparency, no labeling, few opportunities for dialogue and public input. So now we come to the second generation of genetic engineering, which is upon us today and has been upon us for the last maybe decade or so, and maybe we're even in the third now. And I think this is where we place microbiome engineering. And so in particular, I think in situ microbiome engineering is going to be controversial in the sense the ability to add genetically modified microbes to engineer the microbiome, either for human health or animal health or conservation, or to add, like I said, the CRISPR machinery directly, or to introduce it with phage systems, bacteriophage systems. And there are proposals in the literature, as you are much more aware of than I, to use this for animal health, for coral reefs even, to engineer the microbiome in coral reefs, and for plant health. So I won't belabor that point, given that the experts are in this room. So I think here we might have a perfect storm where many of these microbiome engineering applications have a great deal of promise and benefit, especially in the medical realm. But we have a lot of uncertainties as to the function, the stability, the dynamics of these systems, so within the microbiome, but then how the microbiome is interacting with the broader ecosystem, the organism, and even the environment writ large. And so this is just a paper with a lot of outstanding questions on the research and data that we don't have. So in addition, our regulatory system, the coordinated framework, was not designed for microbiome engineering. It was barely designed for genetically engineered animals like fish, barely designed for genetically engineered insects, which we're still struggling with how to cover. So oversight for convergent applications of microbiome engineering, for probiotics, we're really not sure. I mean, for probiotics, it could be a dietary supplement, which doesn't have a whole lot of pre-market oversight. It could be a medical food product, or it could be considered a human drug, which does have a high level of pre-market oversight. So you could go from very little to no to very rigorous pre-market oversight. But what about other consumer products, toys and microbiomes and other things? What about for conservation applications like coral reefs? That's something that, despite the complexity of the microbiome, affects all areas, but in particular, the coordinated framework is not very good at applications for environmental conservation, like coral reefs and such. And then mobile genetic vectors, like the phage delivery or the CRISPR machinery delivery. So basically, everything other than pesticides, industrial microbes, plant pests, and people who claim to have a drug claim, so to actually cure a disease, because a lot of regulation is based on the claim of the product, it's going to be a very uncertain context that we're operating in. Furthermore, I think some of the governance questions for the microbiome are going to be even more interesting than for regular genetic engineering. One might be privacy risks. Lawrence just talked about how we might be able to monitor exactly what you're eating with your microbiome, which can be a good thing if you're trying to form associations between diet and microbiome health. But for privacy, it could be something that you don't necessarily want your doctor or your insurance company to know exactly what you're eating all the time. Furthermore, the microbiome is shared, right? We can transfer it between people, animals to people, environment to people. And so the idea of cross-contamination, of a therapy for one person may be a harm to another. For example, we want to boost the immune system of a patient, but that might get transferred to somebody who has an autoimmune disease, who does not want their immune system boosted. So I think that risk-benefit distribution is going to be another real interesting factor. And then the scope of who do you engage and who do you ask for consent? If the microbiome can be transferred all over the place, who do you have to ask for consent when you put a microbiome out there, either in a public health setting or an agricultural or environmental setting? And then respect for other species. I mean, do we have an obligation to protect other species if we modify our own microbiome for our health? How might it affect species in the environment? And then you have all sorts of the other societal implications that are out there for any emerging technology, access and equity, power and voice, and how do you convey uncertainty to a lay audience? A couple studies, and in closing, I'll talk about a couple studies that I came across on microbiome engineering and public perception. One looked at patient attitudes, so patients that had GI tract illnesses, and consumers may expect more rigorous regulation in the future while simultaneously wanting low costs, easy access, and low involvement of pharmaceutical companies. So there's that public trust component again. Our findings suggest that the current federal regulatory bodies and processes in the United States are viewed with deep skepticism, and that trust, transparency, and the inclusion of stakeholders will be key to the translational process of new probiotic therapies. And then it said, although participants voiced significant doubt of government regulators, they felt that products containing genetically modified probiotic strains should have oversight comparable to that of pharmaceutical drugs. So in other words, in this study, which was based on focus groups and about 130 people, so it's not representative, but they thought that if the probiotic was genetically modified, it should for sure have mandatory pre-market oversight, whereas with some of the other probiotics, they weren't as concerned about pre-market authority. Another study looked at this factor that we call disgust, or the ick factor. You might have heard of, I don't like GM foods because they're ick. Who wants to eat a genetically modified cow or beef? So this study did a very sophisticated study looking at this issue of disgust sensitivity and how it affects public attitudes towards engineering the microbiome. So they had a couple different conditions. They had human versus animal versus plant versus environmental microbiomes. And they had they used the terminology fecal transplant or transplant therapy. So they tried to use terms that might elicit disgust, like fecal, versus terms that would not. And they did find, as expected, that for the human microbiome modifications, genetic modifications, elicited more feelings of disgust. And that the disgust language, when related to the human applications, led to the highest perceived risk. So that's just things that you might expect, but somewhat interesting, because this was the first study I saw specifically on the genetically engineered microbiome. So I've suggested that we take a middle ground approach to governance of emerging genetically engineered products, including microbiome engineering, where it's not all about the science, but it's not all about values either, because the science is important. I used to be a biochemist. I used to make I used to do molecular biology and make genetically engineered plants myself. But there are a lot of models in the literature for kind of these middle ground processes where the science matters, but so does the public values and interpretation of what they care about, what endpoints they're concerned about, and what they consider to be safe. And one of these models is called responsible innovation. So I just want to present it out to you. There are 4 components of responsible innovation. One is anticipation, the need for science to seek to anticipate its impacts; inclusion, the need to deliberate and reflect within an inclusive array of stakeholders; reflexivity, which is the scientists themselves continuously reflecting on their own assumptions; and responsiveness. After you gather all this public input, you reflect and you anticipate, you need to build that into your technology as you're developing it. I have done some anticipation type studies for food nanotechnology back when it was first arising in 2006. I'm running out of time, so I'm not going to go into it. But essentially to scan what's going on in the scientific world long before it comes to market, and then to have a public and stakeholder engagement process to look at different case studies in order to say, is our oversight system prepared? What might be the risks and benefits, and what might be some of these other broader societal concerns that we care about? We've also done it for synthetic biology about 11 years ago, when it was also kind of becoming more and more popular. And so my proposal is that microbiome scientists and engineers work with social scientists and humanities scholars to engage the public and stakeholders in the development of oversight systems, to help design systems prior to market introduction, long before they're introduced into the market, to incorporate public values into the development and decision-making. And this will hopefully enhance, it doesn't always guarantee, but it will enhance trust and might enhance public confidence in science and scientists and engineers. So I will end there. And thank you. And this is our center. We're right down the road at NC State. And we have a lot of people working on the microbiome over at NC State in the agricultural and environmental areas. So thank you again for inviting me.